Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is William Duig from Business Review, and I'll be your host. It is our pleasure to have Capsugel with us today, who will be discussing API-in-capsule, a smart approach for rapid drug development. Today's guest speakers are Mark Capucci, Team Leader of Pre-Formulation and Formulation Development Services at Xcelodose, Ted Kuntz, Vice President Operations at Xcelodose. We also have Catherine Lehman, who's Senior Manager of Marketing for Capsugel Europe. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you do disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send these in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click Submit. We will allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to provide some feedback, and you can do this during or immediately after the webinar. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Catherine.
Okay, thank you, William, for this introduction. And good afternoon and good morning to everyone. So I'm Catherine Lehman, and it's my pleasure to welcome you to this webinar dedicated to API-in-capsule technology and its benefits for rapid drug development. Addressing this topic looked to us at Capsugel particularly accurate, as we know that pressure on time for NCE development has been rising these last years. And this is why using technologies such as precision dosing that is faster than traditional approaches, as we will see, can certainly help to speed up the process to bring a drug into first-in-human. During this webinar, my colleagues Ted Kuntz and Mark Capucci will walk you through the key advantages of the API-in-capsule approach, our experience and offering in this regard. As a starting point, let me quickly introduce Capsugel in a couple of words. So Capsugel is specializing in design, development, and manufacturing of a wide range of innovative dosage forms for the biopharmaceutical and consumer healthcare market. We stand apart in the industry as we offer a unique combination of science, engineering, formulation, and capsule expertise. And this helps us to provide unique solutions for our customers. Actually, we have integrated capabilities from early drug design and development through the commercial manufacturing. And actually, we serve, as we speak, over 4,000 customers over the world, and we do have technology research and product development on 3 continents. That's Capsugel in a nutshell. If we look now and turn quite quickly just to a quick overview about our major technology platforms, I'm happy to say that we have developed unparalleled capabilities in several of essential industry areas. The first of them is definitely everything that concerns technology about the bioavailability enhancement, where we have most probably the most comprehensive breadth of technology on the market, which is particularly anchored in our spray dry dispersion technology and lipid-based formulation know-how that we have in-house. The second pillar of our platforms and know-how is dedicated to targeted delivery, where we use a full range of formulation approaches, including proprietary, multiparticulate, and tertiary capsule technologies, for example, to meet the target product profile of our customers. The third pillar is dedicated to capsules and encapsulation technologies. And here we have really a market-leading capsule range designed to address any application for powder, multiparticulate, semi-solid, and liquid drug delivery. The last part of our know-how is really concerning some specialist solution that we have, for example, in fields like inhalation application or also for pediatric developments. Regarding product development, we employ an integrated model that reaches from early feasibility and best-in-class product design through clinical and commercial manufacturing. Our API-in-capsule and microdosing capabilities that we will discuss today are part of the range of services we offer to our customers for early phase human studies. I turn it now to Ted to walk you through these technologies.
Hi, good afternoon and good morning, everybody. My name is Ted Kuntz. I am the VP of Operations here at Xcelodose in Tampa, Florida. I'll get right moving on the slides here. The two systems that we employ here at Xcelodose, as you can see on the screen, the Xcelodose 120S and the 600S. The 120S is typically used for preclinical evaluations and early R&D studies in the analytical lab. The fill range for both machines ranges from 100 micrograms up to about 100 milligrams. The high-end capacity is much higher than 100 milligrams, but typically, based on the bulk density of the powder and the volume of the capsule, we haven't been able to achieve much more than 100 milligrams on most of our API fills. The typical throughput for the machines, on the 120S, you can see it's about 60 to 100 capsules per hour, and the 600S is much quicker. Because it's an automated machine, it can get up to about 250 to 300 capsules per hour. Again, those are all going to be dependent on the flow of the API. The 120S is a semi-automatic machine. It requires extensive operator interaction. It's manually loaded. Capsules are uncapped, filled, and then recapped by the operator. And then the Xcelodose 600 is it's a fully automated machine. It requires minimal operator interaction, essentially maintaining the empty capsules and the API hoppers filled. So as you can see, the difference between the 120S and 600S is quite dramatic as far as operator interaction. Comparing the Xcelodose versus typical capsule filling, especially in the clinical arena, many times our customers have a lot of questions regarding yield, comparing typical capsule filling issues to the Xcelodose, and it's very, very different. As you can see, I've thrown some considerations up there, and your traditional responses versus the Xcelodose results. Yield, as you all know, when you're doing traditional capsule filling, it's almost nearly impossible to achieve 100% yield. With the Xcelodose, if you set out to fill 1,000 capsules, 5,000 capsules, 10,000 capsules, we almost always achieve that number. The reject rate for traditional filling is always a high concern, and for Xcelodose, once the machine is dialed in and filling properly, the reject rate is almost never a high concern. Blend flow for traditional capsules is extremely important. When the blends are designed and formulated, that's one of the main considerations. But for Xcelodose, blend flow is not important at all. We can always get around the flowability of the API or the blend. The capsule fill weight, for traditional filling, of course, we always rely on gross capsule weight, so the weight of the capsule is always factored into the total weight. This can lead to errors in potency and content uniformity, but with the Xcelodose, it's always absolute net fill weight, and these net fill weights are captured on the data sheets provided. For traditional filling, low weight fills are very difficult. Technology has improved in this area. However, for high capacity microdosing, it's still very challenging. But with the Xcelodose, of course, we can get down to as low as 100 micrograms consistently. And for higher weight fills, traditional capsules always work better. Xcelodose, we can still achieve higher weight fills. However, it does result in slower operation. And then, of course, throughput speed, traditional capsule filling is higher, and Xcelodose is lower. All right, so an overview of the API and capsule. Essentially, API and capsule is precise dosing of very small quantities of an API, a powder blend, or other dosage form into the capsule or other container. And as mentioned earlier, we can get as low as 100 micrograms dosed accurately with RSDs, typically less than 2%. If you notice the picture there, it's very difficult to see, but almost not visible with the naked eye, the amount of cotton to the capsule.
Thank you very much for that, Ted. Okay, audience members, it's now time for the first poll question of today's session. As you can see in front of you, the question is, have you used API in capsule in early stage development work? And the potential responses are yes or no. So just to give our audience a little bit more time to vote, Ted, perhaps you'd like to comment on the question and the kind of responses that you're expecting to see. Ted, are you there?
Yes, Will, I'm here.
Yeah, so I'm just wondering if you could take a couple of seconds just to talk a little bit about this question and the kind of responses that you are expecting to see from this question.
I think we're most of the people that are joining the webinar, I'm expecting to see that they haven't used API in capsule and are interested in possibly using it, and all the different applications that we can apply using this technology.
Brilliant. Thanks very much. So I think we've now kind of given enough time for most people to have submitted an answer if you'd like to continue with the rest of the webinar.
All right, thanks. Okay. So API in capsule benefits, what are the benefits? Obviously expediting and streamlining product development. It increases the efficiencies via precise dispensing of the drug substance into the capsules, and provides detailed comprehensive documentation, including the individual net fill weight of each capsule produced. It's also very cost effective. As we said, the cost to develop a product using API in capsule is very limited because it's essentially the API and the capsule itself. The value, of course, it increases the speed to clinic. A program can get developed very quickly and get phase 1 studies ongoing very quickly, and also proof of concept for various APIs. It can reduce the product development time by approximately 13 to 17 weeks or more. You can have a team developing an API in capsule process, producing material, getting it to the clinic, while simultaneously the formulation development group is developing a tablet or capsule blend, performing stability studies, excipient compatibility studies, et cetera, and not waiting for those results from the clinic to move forward. We have developed the best practices here at Xcelodose. We have over 11 years of operational experience. The first machine was placed in service in 2005 here in Tampa, and as you can see, we've produced more than 600 batches, and we've operated with more than 200 APIs. Each of these APIs has a very diverse array of particle size distribution and flow properties. We haven't seen any APIs that we haven't been able to fill yet. Some key considerations for API assessment. Again, particle size. This can be very important if we're doing very small fills. If you're targeting a 100 microgram dose in your capsules, having large particles in there can affect the dose. Obviously, if a large particle is added to the capsule, it can increase the weight beyond the acceptance range. So it is very important for low dose filling. Particle size distribution, again, extremely important for smaller doses, or if the API is limited. And again, batch yield will be affected. Oftentimes when it is a very low dose, the loss of API is negligible because it's such a low dose. However, it will affect your batch yield. Poor flow can result in high weight variation, especially for lower doses, and increase your reject rates. But again, flow of the API has typically not been an issue for us. Fill weight and volume. The method of filling of the powder greatly reduces the bulk density. It essentially acts as a salt or pepper shaker, and as the API is added to the capsule, the bulk density is reduced greatly. So it does restrict the total amount of API that can be added to a capsule. That's when I spoke earlier about the upper limit of 100 milligrams, that's typically where that comes from. And but the method of filling does help with dissolution and bioavailability because the powder is loosely packed into the capsule. When the capsule is added to the body or dissolution vessel, it is readily available. So some API improvement options, things that we have done operationally when if we experience any issues with the API during filling, we add sieving or screening steps. This is frequently used as a practice here. We would pass the API through a small mesh sieve prior to adding to the hopper on the Xcelodose. And this just simply helps improve the particle size distribution, helps with delumping, helps with flow. Other methods are we have used a mortar and pestle at times. If an API is received in a needle form, oftentimes we can break down the needles into smaller particles and improve the flow and the distribution. We've used other methods such as milling, co-mill, Phipps mill. Those have helped in the past. And again, the Xcelodose parameter settings, tapping frequency, dispensing heads, other methods can be used. There are rare occasions when a high dose fill is used and the flow of the API is very poor. We have been able to manually stir the powder inside the dispensing head and still achieve the dosing weights desired. These are extreme cases and require more interaction by the operator. However, they are successful. Some cases we can add a flow enhancer in small concentrations. We've done that before. It does require more excipient compatibility studies, more development work on the front end. However, it can be effective in improving the flow. We've also done some dry granulation via roller compaction, simply adding the API to the roller compactor, compressing it into ribbons, and then milling it down to larger particle sizes to improve the flow. That's been successful in the past as well. Some other key considerations regarding dosing range considerations. The majority of the dosing ranges are from 1 to 50 milligrams. Again, the smallest dose to date is 0.1 milligrams or 100 micrograms. The largest dose to date we've received is 250 milligrams. However, again, that is a rare case. Most of the doses kind of plateau and top out at 100 milligrams. Again, concern with large doses is the bulk density. Low bulk densities will not fit into the capsule as desired. A good example here is we had 125 milligrams of API and it wouldn't fit into a size 00 capsule just simply due to the bulk density. The RSD of the capsules is normally less than 2%, and it can be slightly higher on very low weight fills. But typically the RSD is very tight and below 2%. And the weight acceptance limits we use is typically plus or minus 5%. We work with our clients. They might specify that otherwise. For lower dose fills, sometimes we broaden the range. And for a higher dose fill, we sometimes tighten the range. Will, back to you.
Thanks very much. Okay, audience members, it's now time for the second poll question of today's webinar. The question, as you see in front of you, is have you considered API in capsules? And the two potential responses are yes and no. So please select the response that's most appropriate for you and click Submit. I'd also like to draw your attention to the resource list widget today, where we have actually got a file about microdosing included in that. So if you would like to go in there, that's free for you guys to download. So just download that, check it out. I'm sure that you'll find it quite useful. However, just with regards to this question, do either of the speakers have any comments on what they're expecting to see or any kind of ideas as to where the audience will be with this?
Yeah, we're hoping that the audience will it'll spark some creativity and some options as far as their drug development needs. There's quite a bit of you'll see on some later slides. There's quite a bit of variability and other applications we can use with this technology. So we're hoping this will spark some ideas with our potential clients.
Fantastic. Okay, so I think we've given the audience quite a long time to vote now. If you'd like to continue with the rest of the presentation.
All right, thanks, Will.
Good morning and good afternoon. This is Mark Capucci, team leader in the Pre-Formulations Formulations Group. Basically, continuing on with some of the key considerations, that we'll see with the API while we're working. A lot of times you'll see that you'll have some environmental considerations of the API while you're working in the area. As Ted mentioned before, we're placing the powder into a hopper that's using a gravity feed system. So some of the things that we do have to take in consideration for some of the APIs is for light sensitivity type compounds, we'll do a low light type of application in the room. We'll be evaluating that in the initial part. Sometimes that information will have come to us. Sometimes that is determined while we're running our early analytical type of evaluations of the API. Some of the other times that we'll see is we'll have some issues with the material. Some of the things that we do in the pre-formulation area when we get some of the material in and start doing the API characterization, where we're looking at the physical and chemical aspects of the API, and then we'll see that using vapor sorption, that some of the materials will have to be handled in a low humidity type of condition. So we'll look at making the room into a low humidity type of environment. We do have low humidity rooms here on the experimental side and also on the manufacturing side. So if the the API itself needs to be maintained at a low level, we can bring it into those low humidity rooms and be able to move forward with that. Humidity, as we know, which will change the aspects of the API, whether it will change its chemical form or some of the other aspects that we have seen is start to have the material become wet in nature and start to reduce the flow, and that's where you'll start to see a lot of the sticking and then a lot of the other aspects that are happening of the API while they're in the hopper. Potent compounds, we do deal with potent compounds here. We do have classification that we currently use for our materials. We have a cutoff of what we can currently use for our system, but if it needs to go into any aspects, we could use some dust protection for the actual process, not only for the API itself, but for the operators to make sure that they're working in a safe environment. Some of the other aspects that are in consideration are the capsule sizes. As you can see, we operate the instruments, the 120S and the 600S, with capsule sizes from size 4 all the way up to size 00. We have currently used gelatin and VCAP Plus capsules here, and as mentioned down in the next bullet point, that basically our Primmel facility has successfully used and filled and closed using sprinkle caps as well. Some of the other things that were mentioned before when you're looking at API in a capsule is the parallel studies that can be going on in the formulation area while you're moving forward with just your API in a capsule, and some of the aspects of that Ted mentioned before, that you can do your excipient compatibility studies in parallel with moving forward with the assessment of just the API in the capsule. Some of the key considerations when you're working with the 120S, this would be the instrument that's in the experimental area. This is where we'll be getting the API firsthand. A lot of times the API will not have been certified yet, so we'll have the ability to work with the powder and look at all the characterization aspects of it and be able to determine what would be the correct dispensing head and also the correct capsule size. So this is where we'll be going in and doing pretty much method development, setting up parameters for the instrumentation, so that way when we pick a successful dispensing head, dispensing time, and capsule size, that can be transferred over to the manufacturing group, and that can be written into the batch record and be able to move forward with the process. As some of the aspects that we see that change moving the process from the experimental area to the manufacturing area, as listed, when material will come in, we want to make sure that we're working with the same lot that will be used for the GMP campaign. And some of the aspects that change that are the ones that are listed below. If we're working with an experimental lot to the GMP lot, particle size distribution, powder flow density, and morphology of the material can drastically change the fill of the capsule and the flow of the material. And that's some of the parameters that we'll look at in the beginning to have a good determination of what we're going to be using for the dispensing head and everything else. So as mentioned before, the capsules are produced for the various aspects to be able to start, at least from an experimental point of view, and be able to get the process moving. All right. Basically, we have a couple slides that will be coming up. Will be a couple case studies that we've had here at Xcelliance, keeping it very general to be able to at least show some of the range of some of the parameters they had to work around. The first case study that you see listed on the slide is in regards to a low fill. We had a client that came to us that had a highly potent drug that needed to have for low dose study. Basically, the amount that needed to be dispensed was the 0.1 mg fill volume into a size 1 capsule. The biggest constraint that we had with the project was to be able to complete the run with a plus or minus 5% weight range. And as you can see from what is listed on the slide, that window of acceptable capsules is very small. So we were going through several evaluations with different dispensing heads and tapping frequencies to be able to produce capsules that were within that acceptable range. And moving forward, because as Ted mentioned earlier, when we start working with lower fills, we start to open up that range a little bit to somewhere around 7 to 8%. For this particular API, we were successful to be able to give the client the product and be able to move on with the clinical trials and keep the yield of the campaign in its successful aspect. Case study 2, this was unique for us. The Xcelodose works in an aspect where it's filling a capsule, and basically that fill weight is going to be generated into your run report and your batch reports, and then that's basically the drug product itself. In this particular case, since we're working in the experimental area, the client wanted to actually fill in 2 components into 1 capsule. As looking at other aspects before we even started to use the Xcelodose were the other aspects of flood filling, weighing the capsules, making a blend, et cetera, et cetera. Unfortunately, all those aspects were not viable to the client to be able to move forward, so came up with a decision to basically use the Xcelodose for a 2-component fill. The capsules were filled first with the 1 component, the inactive component, to be able to move forward, fill those at a rate that was determined based on the molecular weight of the API, and be able to go from there. The API that was being filled in the capsules has 3 different strengths, so that's where a blend wasn't able to be used to be able to move forward with it. So the 1 component would be filled in the capsules. Those capsules were capped, loosely set off to the side, and then we put the capsules back onto the Xcelodose and filled it with the API, and be able to show with the 2 batch records the 1 fill of the 1 component, and then coming back around and being able to fill the capsule itself with the API and be able to move those forward and give those to the client. And then the studies that were showed for the results were favorable to be able to move forward.
I just want to jump in here and clarify that with case study 2, which Mark was just describing, that was done on the 120S, which is the semi-automatic machine, lots of hand opening and closing of capsules. It's not capable to do a 2-component fill on the 600S machines.
Thank you, Ted.
Sure.
Okay, I'll hand this over to Ted.
All right, so thanks, Mark. Getting back to it, so case study 3, we had an interesting challenge here in Tampa. We have a client we're working with They have a pediatric dosage form. We developed 2 different pellets, and we had a wide range of dose. It was about 7 dose ranges for this drug product, and 2 or 3 of the lower dose products had very small doses for one of the pellets. So, for example, there I'm showing it had 7 to 8 pellets for a dose. We were looking at using a sachet to deliver this. The client wanted to pour it onto soft food. When we were dosing it into the sachets, we found that the drug would, the pellets would hang up in the creases of the sachet, and with a 7 to 8 pellet dose, 1 pellet loss would reduce the dose by as much as 12.5%. So obviously we had issues with delivering the dose accurately. So what we did is using the capsule concept with the 120S, we could dose 1 pellet 10 times, then come back and dose the second pellet into the capsule 10 times. Sorry, when I say 10 times, that's the number of capsules that we can fill each time on the 120S. But essentially, we were able to do this proof of concept using the sprinkle cap, and the client was very pleased. It took the whole logistics and negative aspects of the sachet out of the equation, and you can use this concept using a high-speed capsule filling machine as opposed to a sachet filling machine, so the accuracy of the dose was increased there as well. So the use of the 120S and the sprinkle caps really brought this project together, and it made the client very happy. It took a lot of challenges out of the mix. So very excited about that. So moving on, case study 4. I apologize for the header there. There's a misspelling there. It should be filling of formulated blends. It says beads right now, but essentially we had a client. We had done a full capsule formulation development project for them, very successful. They came back to us and they were very desperate. They said, we need to do a pediatric study. We started talking about a full development project, and it was going to take weeks and months and lots of high costs. So we talked to the client. We said, look, let's just take the blend that we've already made. Let's dose it down to about a 33 milligram fill using the Xcelodose, and we don't have to reformulate. We just use the same formula and move on from there. So the client was very skeptical, but the method of filling with the Xcelodose, there is quite a lot of vibration and tapping in the dispensing head, but the dwell time in the dispensing head is fairly short. And if the particle size distribution of the blend is fairly uniform, no big disparities, the blend uniformity shouldn't be an issue. So we proceeded on with this project, and we filled the capsules using the Xcelodose 33.3 milligram fill to get a 5 milligram dose for pediatrics. The client was very happy. The results couldn't have been any better using any other technology. As you can see, there's no segregation, no issues with assay or content uniformity. We saved them a ton of time with development work and money, and the CTM was delivered on time. So that's one of our favorite success stories using the Xcelodose. And then another case study, case study 5, talking about manufacturing capacity. Now, as you guys can see, the throughput with an Xcelodose is not very quick. It's not very fast. We've said 250 to 300 capsules an hour on a good day. But with the manufacturing capacity here at Xcelliance, we have produced batches as big as 75,000 capsules. The fill weight was 50 milligrams, which is fairly high in the range. Actually, it's about mid-range for the Xcelodose, but we use size 0 capsules. And we were able to produce these batches in 4 weeks. Now, again, Xcelliance has 6 Xcelodoses in operation here, so we dedicated 1 Xcelodose to this batch. Again, the batches was done in just under 4 weeks. Something happened there. And all things considered, it is a relatively short production time using this technology. The client was fairly amazed. We've done work for them in the past, and a batch that size didn't seem conceivable. But we were able to produce this because we have manufacturing operations here at 24 hours a day, 5 days a week. We have 3 shifts of operation, and with the acquisition of Capsugel earlier this year, we have a very good access to change parts, and our machine downtime is very low. So much success there. Case study 6, we've had some more unique applications similar to the powder blend. The client wanted to perform some comparator studies. Often if we do comparator studies, we would just use an Ultra A type machine, add the capsules or the tablets into the capsules and blind them using a capsule. But they wanted to do comparator studies using various ranges. So they came to us and they said, hey, we need some lower doses. We can't just use the 250 milligrams and the 400 milligram tablet. So we were thinking outside the box, and we said, let's mill these tablets down into a powder, and let's fill that powder into the capsule using the Xcelodose and get the doses that we need. So as you can see, we used the Quadro co-mill. We passed the tablets into the co-mill, blended into a powder, and we were able to adjust the fill weights to achieve 80 milligrams to deliver 50 milligrams of the drug substance. Again, very good results with content uniformity and assay, and the CTM was delivered on time. The client was very happy with this. It gave them a lot of flexibility with what they were trying to do. All right, Will, back to you.
Thanks very much for that. Okay, audience, so it's now time for the final poll question of today's session. And the question is, would you agree that time is the critical parameter in early development stages? And the potential responses are yes and no. I would also, again, like to take this opportunity to remind audience members to submit some questions for our live Q&A session that's going to be taking place at the end of today's session. If you want to submit a question, just type your question into the box at the top left-hand corner of your screen and click submit. But in the meantime, perhaps Ted and Mark would like to comment briefly on this question.
Yes, we have seen that time is a critical path for a lot of the clients. A lot of times API will come in and basically want to be able to move forward with at least getting some initial type of information, whether it be a feasibility study, getting material into the clinic, just to see if the drug itself is going to be able to move it through its life cycle. We have seen in some times where it is successful, and unfortunately some other times where some tweaking will have to be done of the drug substance itself, or unfortunately where some of the projects at that point are terminated due to the fact that the results from the clinic are not favorable to be able to move forward. So having API in a capsule is very helpful to eliminate time that is needed to go into a full tablet type of formulation.
Brilliant. Thanks very much for that. Okay, I think so we've given quite a lot of time to answer the question, so feel free to continue with the rest of the webinar.
Thanks. Katrin, I think we're going to pass it over to you at this point.
Yeah, I just have some comments on the European capabilities. Do you want to comment on the Tampa capabilities, Ted or Mark?
Oh, sure, I can do that real quick. Sorry about that. Just to rehash, at our Tampa facilities, we have one Xcelodose 120S in our R&D area, and we have 4 Xcelodose 600 systems in our GMP area. It lists high containment there. That is a typo. Xcelliance in Tampa does not have high containment, so we don't use isolators for our machines. We use flow hoods. We can handle potent compounds, but not highly potent compounds. Again, the analytical services that we offer here, method development, technical packages for APIs, cleaning evaluations, stability program management, and the pre-formulation services, as Mark talked about, powder-in-capsule, powder-in-bottle, powder blends, what is MP tablet? Sorry.
Yes.
Single bilayer compression, oral liquid suspension, semi-solids, and we have been FDA inspected several times this year. So.
Okay, so just to complement this picture that is for the sample site, for Europe, actually our center of expertise is in Ploermel in France, where we have actually two Xcelodose 600 running, and beyond what we can offer in terms of GMP microdosing capabilities, we also offer some analytical services such as method development and validation, ICH stability studies, that allow us to support early-stage projects up to clinical programs. Aside the simple API in capsule approach, the Ploermel site also offers formulation services looking at a wide range of solid, semi-solid, or liquid-filled hard capsules. And as well, by the way, it's not mentioned here, but enteric drug solution are also in the scope. Looking at the regulatory status, in Ploermel we have also been FDA inspected, but additionally the site has also been inspected by the European agency and the Brazilian agency as well. So this is summarizing our microdosing services, centers of excellence across the globe. Maybe just turning now to the last part of the webinar, which is basically looking at the key takeaways that we can have after this session. I think the first point is definitely that I think we have reviewed that the API powder-in-capsule or the API-in-bottle approach is really a key tool to rapidly carry out proof of concepts or first-in-human studies, especially because they allow to do this without extensive formulation and compatibility study, and thereby speeding up time to first-in-human. So that's, I think that was pretty clear. In terms of technologies, the Xcelodose equipment is best in class for microdosing, and I think Mark and Ted worked us around the different case studies that were presented that are really demonstrating this. I think you also understood that Variance is the best practice in Tampa, has the best practice in terms of microdosing technology, and this has been now extended across various sites of Capsugel, and including the Ploermel site for Europe. And finally, as mentioned before, and as in the introduction, Capsugel is also offering a full range of solutions beyond microdosing that aims to support new product development from design stage to the manufacturing at industrial scale. I think this is closing our session. I think on the slide here that we'll be showing now, you will see the different contact points. So we have our two speakers, Ted and Mark, and additionally we added also contact for potential to reach out to our European facilities over here. And I think we can then start the question and answer session. Okay, maybe just starting the questions that we have here, Ted and Mark. What I see here coming from Boris Gorin is a question about, which is the following: how does PSD of API affect the dosing consistency? I don't know who wants to take the question.
Okay, basically particle size distribution is important. We have seen in the past where we've mentioned we've had to use some unique operations to be able to get the particle size down into uniform nature. That will help with flow through the dispensing heads. The dispensing heads themselves are set up that have a number of holes that are there, and the diameter of the holes are set. So if you get the particle size down into a uniform nature, and along with the functionality of the gravity feed system, it helps with the flow, it helps with filling the body of the capsule, and helps with keeping all the capsules within an acceptable range to be able to move forward.
Okay, great.
Move on.
Another question that came in, thank you for this. Another question that came in from Nico Mel, which is the following: how do you handle API batch-to-batch variability, especially in terms of bulk density?
Yeah, good question, Nico. That is frequently a topic of conversation with us. It's in early stage development, as most of you know, API process development is very difficult at the onset. So frequently we will get a batch of API that performs one way on the Xcelodose, and then the next batch will be, chemically it'll be the same, but physically it will be completely different. So we have enough experience with the Xcelodose technology that we're able to adapt the parameters and still complete the batch and produce the amount of capsules that the client needs. So it is an issue. We see it a lot when it comes from Mark's area over to the GMP area, from the R&D side over to the GMP side. You know, the client will send an API, and it works on Mark's side, and they get some parameters, and then we get something that's completely different in GMP. Yeah, we have to adjust the parameters and make some tweaks, but we'll be able to produce capsules and achieve the goal in the end. So we see it a lot, but it's typically not a challenge for us.
And to add to that, what we suggest is when we're working with the initial API that's coming in and it hasn't been fully classified, once that material has been classified, or if the parameters have been changed, or any of the chemical makeup of the API has been changed, is we will receive it back in our area again and at least evaluate it from what we did initially to make sure that it's at least flowing in the same respect, handling it in the same respect, and then that transfer over to the manufacturing group to make sure that when they're ready to start their campaign, they're not seeing any major issues and they all have been taken care of ahead of time.
Okay, thank you. Yeah, there was another one. Could you please clarify if the 120S at Xcelodose can also be used for GMP manufacture or just technical?
The 120S that we have in the experimental area is used just for that. We don't use the 120S for any GMP manufacturing campaigns. Anything that needs to be in basically a GMP environment will be in the manufacturing area, which will be using the 600 and the 600S.
But to clarify, the 120S can be used in a GMP environment. It is in use in various GMP environments over the world. It can be. Here at Xcelodose, we use it strictly for R&D, but yes, it is used in a GMP environment at various spots across the world.
Okay, another question. What kind of stability program is required for phase 1 study in case API fill into hard gelatin capsules without any excipients? What is the sampling points during the stability, months or days?
For feasibility studies using API in a stability sense, we have typically used two different types of conditions, your 2560 and 4570, and a lot of it is client driven. We have seen some studies where it's just API and capsule that have been extended for 2560 all the way out to 24 months. And then we've seen some other stability studies with direction of the client and in conversation with the client that will end somewhere around a year at 2560. A lot of times what will happen is when we generate the material in the experimental area and we'll place it up on stability, we're usually getting about one month for at least on a risky type of approach, up to maybe 2 or 3 months of getting some stability data, and then the decision is made to move the material into the GMP area to start the GMP campaign. And so that way you're having 2 studies that are going on at the same time that are bracketed, pretty much with a couple months apart from each other. So that way you're still getting your information initially and then being able to have that data and be able to move forward. So it really is determined by the client and also at a more of a conservative or risk type of approach as to how much data is needed before moving it on to the next step.
Okay, another question is about is the former UK NCAP site also having Xcelodose, or is it all at the Pluramel site? So I'm happy to answer this one. So basically, our capsule site in Pluramel is the center of excellence for our microdosing capabilities for Europe. So all the capacity has been transferred to the Pluramel site. Just another question regarding to the formulation for amorphous solid dispersions via hot melt extrusion. What method do you use to grind the extrudates and control the particle size? That's the first question. And the second question, is the extrusion temperature always be higher than the API temperature?
I'm sorry, I can't answer that question. We do not do hot melt extrusions here at Xcelodose in Tampa, so I cannot answer that question.
Okay. Another question, do you measure any stability of the API in soft food?
We would not perform those studies here at Xcelodose. We are limited to producing the drug product and providing the drug product to the clinics. But that would be the client's responsibility to work with the clinic moving on forward to test that.
Okay. Another question, has API in capsule ever been considered for commercial products?
Yes, it has been considered quite a few times. It sounds like a pretty, at first it sounded like a bold approach, but in a rare case, if you had an API that was suitable for that, the drug stability was very good. Using API in capsule for commercial product, it eliminates the potential harmfulness of adding it to an excipient that wasn't compatible to it. So it has been considered. We've talked with several clients. We have never gone forward with it, but oftentimes it comes down to capacity as well, because the Xcelodose is relatively slow throughput. That is a consideration, but it's a viable option. We work with a lot of clients that have niche products, orphan products, rare diseases, where the batch sizes are not very high. It is a possibility. And let me back up there. We do have one client that we are working with there. I saw a question also on the list talking about inhalation products. We do have a client that has a product that it's a lyophilized blend. So they send us the API. We mix the API, get it into a cake form using lyophilization process, and then we add that lyophilized blend to a size 3 capsule, and then the capsules are packaged here in blister package form, and they're used for inhalation and a dry powder inhaler. So that's a very common application. We have low humidity capabilities here. Oftentimes the dry powder inhalers are humidity sensitive, so the capsules are filled in a low humidity environment, typically somewhere between 30 to 40%, 45%. So that is a common application, and thanks for that question. Okay.
Maybe one of the last one we can take, I think, William, is the following. How important are powder flow characteristics when filling capsules using Xcelodose?
The powder characteristics are very important. That's where we'll look at the API initially before we start doing any work on the Xcelodose. We'll have at least an understanding, if it's not provided, of the flow by using basically your typical flow decks. We'll do bulk and tap density of the material to at least have an understanding of how it is with compaction. We'll also look at particle size determination that we currently have here, other aspects looking at morphology, just having a good understanding of what the material is like if that information is not provided, so that way when we're moving on into the Xcelodose, that we at least have some of the information to at least help with picking out the proper dispensing head and then picking the proper tapping frequency. As mentioned a couple times throughout the course of the webinar, the material is sitting in the hopper and there is a tapping arm that is tapping the dispensing head and basically looking at to make sure that the material is flowing properly through that. We're not seeing any bridging, we're not seeing any rat holing, and some of the other aspects to be able to make sure that the material is flowing, to be able to keep the process moving forward. As Ted mentioned, when we start getting into higher fills, the fill time and the tapping frequency has to be changed. So that way you'll be able to get material in a consistent, so that way you can keep the time for each actual fill of the capsule down into at least a good parameter. Some of the parameters that you do not want to have is where it takes 20 to 25 seconds to fill a capsule. That will turn a campaign into a very long time. So we'll work with not only with the flow of the API, but also the parameters of the instrumentation, be able to make sure that we're able to get it down and be able to move forward.
Okay, and maybe the last question, how many capsules can you produce in a day? What is your rate limiting factor for this production?
Speaking from a GMP manufacturing perspective, typically the rate limiting factor is the fill weight. So again, the Xcelodose filling method is converse to a typical capsule filling. On normal capsule filling equipment, you want a higher dose works better. It's easier to control the weight, but on the Xcelodose, a lower dose actually works better. A 1 milligram fill or a 5 milligram fill, it's quicker to fill. If the fill weight's 25 or 50 milligrams, it just prolongs the filling because you're physically sitting there waiting for the capsule, the powder to fill the capsule body. So that's a consideration, the dose weight, and then the flow of the powder is again another one. If you've got a powder that doesn't flow well and the dose is high, it may require some manual manipulation of the powder, which takes a little bit longer. But those are two aspects. And then how many capsules can we produce in a day? Again, we run 24/5. So in a shift, we can typically produce anywhere. I know we mentioned some numbers on the slide, but we can produce anywhere from 1,000 to 2,000 capsules a shift. And that gets you anywhere from 3,000 to 6,000 capsules a day. We typically say we're right at around 4,500 capsules a day because that seems to be the mean of our production, but it can vary. Again, for this concept, typically the batch sizes are down around several thousand capsules to 10 or 20,000 capsules, and we do get larger batch sizes, but we can usually complete a batch within a couple days.
Okay.
That's it.
So I think we are over the questions, so most of them. So thank you all for attending. Thanks Ted and Mark for having put this together. And I think, William, I hand it over to you now.
Thank you very much. Yeah, so that does just leave me to thank Mark, Ted, and also Katarine for it was a great presentation and Q&A session, and also to Capsugel for sponsoring today's session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of today's webinar, or you can access this through our website, which is www.business-review-webinars.com. Once this webinar ends, today's survey will appear in its place, and we would appreciate it if you could take the time to answer the questions provided in there, just to give us a little bit of feedback. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope you all have a very nice day.